Hello, everyone. Welcome to this second session in which we will cover the concept of phylogenetic mutations. So, phylogenetic mutations are, or the concept that we want to introduce is very simple, actually. But for some of you, might sound a bit advanced, or in our experience with our previous editions of this workshop, some I know it was a bit difficult for some people, and I think the difficulty was um, in the fact that you need to understand what a phylogenetic tree is, at least the very basics. But I will try to introduce these very briefly, and I will try to explain it as schematic and as simple as possible. So for our example, we will imagine that we have certain mutations, which are represented here in red color, different shapes, that confer drug resistance, because this is what we are interested in this webinar about the phylogenetic mutations in the context of drug resistance. And then we can imagine another mutation, which is represented by this blue dot, which is a mutation that does not confer drug resistance. And then we have a phylogenetic tree that represents the genetic relationships between samples. This is what a phylogenetic tree means, really. So here at the tips are represented our samples, the ones that we are analyzing, and these are the genetic relationships. So these two samples are genetically more close between them than they are to this one, for example. Now, we are going to talk about something. We are going to say that this blue mutation happened here in the phylogeny, and we are going to say that these red mutations happened here in the phylogeny, and you might be a bit lost so far, but I will explain what this means in a moment. So normally we don't have this phylogeny, and we don't know when mutations happened. So what we have are genotypes, right? So based on the genotypes, these are genomes. We can compare genome to genome, and then using phylogenetic inference algorithms and software, we can infer the phylogeny. And then having the phylogeny, which is the genetic relationships between samples, and having the different mutations, we know when mutations happen in the phylogeny. And what this means, it's very simple. I'm going to explain it. So if we have this phylogeny that represents genetic relationships, and we observe these genotypes, that we know these genotypes have his relationships, and we observe these blue mutations in all the genotypes, well, the most simple explanation is that this mutation, the ancestor of all these 5 samples, have had this blue mutation. This is not the only explanation. Of course, you could imagine that this blue mutation happened here, 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 and here independently. So these 5 bacteria acquired independently this blue mutation, but this is way less unlikely that so the most simple explanation is that actually, because all of them have this blue mutation, the ancestor of all these 5 had this blue mutation. Right, and of course there is no other possible explanation, because if we said, well, what would happen if the blue mutation happened here? Well, if it happened here, then we should observe this blue mutation in all the strains, but we only observe in this, right? So basically what a phylogenetic mutation means is a mutation that is shared by different bacteria or by different organisms, because the ancestor of all those organisms have that mutation. And why is this important, and why is this interesting in the context of drug resistance? So I will explain it in a moment. The problem is that if we would like to identify mutations that confer drug resistance, now, if we don't take into account the phylogenetic evidence, so if we don't take into account the phylogenetic data that we have, we just take into account genotypes and mutations, and we try to perform an association analysis of genotype to phenotype, as we mentioned in the previous webinar, we might conclude erroneously that the blue mutation is associated to drug resistance. Why? Because most of strains that carry the blue mutation are drug resistant. And then we could we could conclude that. But that would not be erroneous. That would not be um, accurate, because the reason they share the blue mutation is just because it's one mutation that they all share, and this is not conferring drug resistance. And why this is important in the context of drug resistance is because there are sub-mutations, like this red mutation here, the circle, that we can observe also in many strains. But in this case, these are not phylogenetic mutations, because the reason these mutations are here is not because they share a common, all of them, they share the same common ancestor with this red circle, right? You see that if we observe the genotypes and we observe the phylogeny, the most simple explanation is that this red mutation happened here, and happened here. So instead of happening only once and then all the descendants having this blue mutation, it happened independently. And this is what we call homoplasy. So in biology, homoplasy is when we observe the same trait. It can be a mutation, but it can be other things that evolved independently. For another example, so maybe you can understand this better, is the wings of insects, the wings of bats, so mammals, and the wings of birds. So the reason these have wings is not because they all share a common ancestor with wings, is because these different groups of animals evolved wings independently. The reason most likely they evolved wings independently is because having wings is very advantageous. It's very it probably increases the fitness of that particular organism a lot. So that's what happened also with drug-resistant mutations. So basically, when we observe that a mutation, not only drug resistance, when we observe that a mutation happened many times independently, that's indicative that there is some strong selective pressure, some strong selective force selecting for that mutation. So that mutation somehow gives a huge advantage to that organism. And you can imagine that drug pressure antibiotics is a strong selective pressure, because when you treat with antibiotics a population of bacteria, only those that have drug resistant mutations can survive. All the ones that do not have drug resistant mutations will die, and then we won't produce any descendant. So these will be the ones that we will observe many times in different patients. Um, and these, again, as opposed to phylogenetic mutations that happened, well, they can happen for many different reasons, but we observe them many times in strains, not because they happen many times independently, not because they are selected for many times, but because they um, strains carrying this mutation are descending for the same from um, the same ancestor. Homoplasty analysis, as you may imagine, is also a very powerful tool to detect mutations that might be or identify mutations that might be related to drug resistance, because basically homoplasty analysis is a powerful tool to identify mutations that are selected and then are providing some advantage to the organism that has those mutations. So then how do we know, or how do we in practice identify these mutations and distinguish them and know which ones are really conferring drug resistance and which not? So of course you are not expected to do this analysis every time. You can do it, but there are other studies already that have done this kind of analysis, identifying phylogenetic mutations in tuberculosis. This is a very nice example. So there are many phylogenetic mutations that are identified in these and other works. So for example, in our laboratory, this is the way we proceed. So we have a list of phylogenetic mutations that we have identified, or that including the phylogenetic mutations in this work. And what we do is every time we detect drug resistance, we check whether any of the mutations are any of these phylogenetic mutations that were detected here, and if so, most likely they are not conferring drug resistance. So now to finish, I would like to introduce this is exactly the same example that I provided before with the blue mutation, but now the mutation is purple. And the reason is now this mutation confers drug resistance. So what happened evolutionarily is exactly the same. And the only difference is that this purple mutation confers drug resistance. And the reason I put this here, this scheme, and the reason I'm mentioning this is because the fact that we call a mutation phylogenetic is not related to the fact of whether it confers or not drug resistance. We call phylogenetic mutations to those mutations, again, I repeat, that are shared by many different strains only because they all descend from the same ancestor. But again, it might be that some mutation, because all mutations can have a biological effect, not only in drug resistance, in many other things. So it can happen that a mutation, or put in other terms, the reason we care about these in the context of drug resistance is because it's not only useful to detect which mutations confer or not drug resistance, in this case all mutations confer drug resistance, is because it's also useful to study evolution of drug resistance. So here the red mutations in this example, they are appearing in the tips of the phylogeny, and there are many homoplaces, or there are several homoplaces, as I mentioned before, and this is because these mutations most likely are arising as a consequence of drug treatment. So there is some antibiotics. So we are using antibiotics, and then we are selecting for strains that are drug resistant. This purple mutation also confers drug resistance, but it happened at some point in the history, but not because we were using antibiotics. It was not because the pressure of antibiotics. And maybe you think that this is a strange example, but actually we have a we have a very good example in the Mycobacterium tuberculosis complex, which is Mycobacterium bovis. So it turns out that we say some of you might have heard that Mycobacterium bovis is intrinsically resistant to pyrazinamide. And the reason we say so, and the reason it is like that, is because there is one clade of Mycobacterium bovis that around 900 years ago acquired a mutation that confers pyrazinamide resistance. Of course, this was 900 years ago, so as you might imagine, this was not because of the use of antibiotics. It was a mutation that happened, and it turns out that today that mutation confers resistance to an antibiotic that we have that we use for tuberculosis treatment. But again, this mutation did not evolve due to the use of antibiotic. It just evolved, and it confers drug resistance. It turns out to confer drug resistance. Um, yeah, so that was all with phylogenetic mutations. I hope you understood. Otherwise, don't worry. There are several concepts that might be a bit misleading here to understand, a bit difficult to understand, but just remember we have the live sessions. You can ask always. We will be there to answer your questions, and see you in the next webinar to talk about tuberculosis transmission and clustering analysis.